Okay, hello friends. This is the lecture on biochemistry, a small refresher, small topic for you about this. We're going in this. We are not talking about the glycolysis. We will be talking about the regulation of the enzyme important for the glycolysis. See, friends, if you know this glycolysis, you are taking a glucose molecule and that you are finally producing a pyruvate, producing pyruvate molecules, and there are certain enzymes in this step which are certain enzyme in this mechanism which are uh, promoting the formation of this pyruvate. So you have some enzymes such as hexokinase. I have taught everything about glycolysis. You can watch my videos, uh, maybe in the drive they are available. So hexokinase, you have some phosphofructokinase, and you have aldolases, you have no isomerases. Okay, so here I am concerned about the regulation of this enzyme. I want to take your attention towards this part. When your when your fructose 6 phosphate is being converted into fructose 1,6 bisphosphate, because I came to know that some students have some doubt, or I felt that I should make a single video on this regulation of PFK enzyme. It's most important enzyme. Its mechanism is somewhat, you may be confused, but follow the track, whatever track I'm telling you to follow. Don't use your own brain. Okay, just follow the track which I am telling you to follow. Then only you can get to it. You can get you can get the knowledge about this regulation of PFK. Now see friends, if I take this sample here, I am extracting this sample and writing here. Okay, I'm extracting this sample and I'm writing it here. Now see guys, see, if I take this enzyme, if I take this whatever I was showing you, fructose, what happens to this? Just a second guys. Okay, fructose 6 phosphate, fructose 1,6 bisphosphate, fructose 6 phosphate changing to fructose 1,6 bisphosphate by the enzyme phosphofructokinase. Phosphofructokinase is, we call it PFK-1. This phosphofructokinase is converting fructose 6 phosphate to fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. Easy. Since it is a phosphofructokinase, it will be using an ATP molecule. It will be using an ATP molecule, and that ATP will be converted. He has taken that phosphate and now it is ADP. It uses, I taught you in the beginning, kinase uses phosphate. The basics of fundamentals of biochemistry, I have already made it strong. If you have any doubt in that, please, your colleagues, they can share my notes. I have explained them clearly how, what is the basic principles and how we are going to apply those principles in the biochemistry. Now see my friends, I am going to talk about this enzyme. See, I am going to talk about this enzyme guys. I am going to talk about this enzyme. This is the beautiful enzyme who is doing all this. Okay, let me go to this enzyme. This PFK enzyme, this PFK enzyme, this PFK enzyme, I'm talking about this enzyme guys. Okay, be oriented. I'm talking about this enzyme. This is an allosteric enzyme. What? It's an allosteric enzyme. What do we mean by allosteric enzyme? That means I can increase its activity and I can also decrease its activity. Its activity can be increased as well as decreased. Now it will depend. If you want to increase its activity, if you increase its activity by an agent, we are using the word, we are using a positive allosteric enzyme inducer. We say positive. If I am inducing this, I say it is a positive allosteric inducer. It is a positive allosteric inducer. And if something is inhibiting it, I say it is a negative allosteric, or it's an allosteric inhibitor, isn't it? We can say it's an allosteric inhibitor. Okay. So negative allosteric, you can use the word. So see friends, what is inducing this enzyme? This enzyme is being induced by a compound that is called fructose 2,6. Let me make it clear. This is being induced by fructose 2,6 bisphosphate. Fructose 2,6 bisphosphate and adenosine monophosphate. If you have more amounts of F26BP in the cytosol, if you have more amounts of fructose 2,6 bisphosphate in the cytosol, that is the cytoplasm, you will be inducing this enzyme PFK-1, and this PFK-1 enzyme will be converting more amounts of fructose 6-phosphate to fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. If you are inducing this enzyme phosphofructokinase, you will be actually accelerating the process of conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. Is it clear? Is it clear? Now I want to tell you a story. This was about India. So who is inhibiting, by the way? If you have now opposite, if this is AMP, this should be ATP. So ATP will inhibit this PFK. Citrate will inhibit. Citrate ions will inhibit this. They will inhibit this. What else? Protons, H positive ion concentration. If you are increasing the proton concentration, this enzyme will get inhibited. This enzyme will get inhibited. Is it clear? Is it clear? Now listen to my words, guys. Now, see, this story has to be in an oriented fashion so that we can have that brain orientation. Done? Now listen to my words. How do we produce this F26BP, guys? How we are producing this F2? Now going to this backstory. Now see what we are going. We took our first step, we took first step back. Now we are taking another step back. Are we able to understand? Now I am telling you F26BP, how is it made? Basically, F26BP is always present in the cytosol. It is produced by some F6P molecules. There are some biomolecules present in the cytoplasm which are getting converted to fructose 2,6 bisphosphate. Fructose 2,6 bisphosphate. And we said fructose 2,6 bisphosphate is itself an inducer of PFK, which is converting the fructose 6-phosphate into fructose 1,6 bisphosphate, isn't it? Guys, isn't it? That's what I taught you in the above. I said, if you want to convert fructose 6-phosphate to fructose 1,6 bisphosphate, phosphofructokinase 1 is going to do that with the use of ATP molecule. Now this phosphofructokinase is having some inducers, is having some inhibitors. Inducers will be fructose 2,6 bisphosphate and adenosine monophosphate. And if you want to inhibit this enzyme, if you want to inhibit the glycolysis, you increase the ATP. You increase the ATP, you will inhibit the glycolysis. You increase the citrate ion, you will inhibit the PFK-1. You increase the protons, you will be inhibiting the PFK-1. Now, fructose 2,6 bisphosphate, where is it coming from? This fructose 2,6 bisphosphate is being formed from molecule that is some fructose 6-phosphate. Fructose 6-phosphate is making what? Fructose 2,6 bisphosphate. Fructose 2,6 bisphosphate. Okay. So this fructose 6-phosphate is getting converted to fructose 2,6 bisphosphate. Okay. Now fructose 6-phosphate, which enzyme is there? Which enzyme is there to convert this? It is phosphofructokinase 2. Guys, this was the confusion I had in my first year, guys. I used to cry on this. Okay, now it's clear. So phosphofructokinase 2 will be converting this fructose 6-phosphate to fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate. And this enzyme PFK2 is only active in the dephosphorylated state. If you dephosphorylate it, you will be activating it. So you do dephosphorylation, you will be activating it, and you will be making more amount of fructose 2,6-bisphosphate, which will be a positive allosteric inhibitor, inducer of PFK1, which will be doing what? Glycolysis. Is it clear? I was having that thing in the mind. That's why I told you to take a short session, because you get confused every time with this concept. Okay, this is a concept you have to understand. Now, fructose 2,6-bisphosphate is converting your is inducing your PFK1, which is converting your fructose 6-phosphate to fructose 1,6-bisphosphate. Can I say phosphofructokinase 2 is responsible, is a side regulator who is basically regulating this F2,6 BP? Can I say that? If I don't have phosphofructokinase 2, I will not be having fructose 2,6-bisphosphate. If I don't have fructose 2,6-bisphosphate, I will not be inducing the PFK1. If I'm not inducing the PFK1, I will be having no conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. I hope you are understanding what I mean by the language. Fine. Now, insulin is active in which state? Dephosphorylated state or phosphorylated? Is it active? Okay, I will be uploading it in YouTube, don't worry. So is it active in deep? Very nice answer. Insulin is active in dephosphorylated state. It's active in dephosphorylated state. And also insulin will be dephosphorylating the enzymes. It will be dephosphorylating the enzymes in the cell. Once you bring the insulin on the surface of the cell, insulin is coming here. The tyrosine kinase activity, what a beautiful concept it is. That insulin is coming at the receptor on the cell will lead to itself is active in the dephosphorylated state and will lead to dephosphorylation of the enzymes present inside the cell. Will lead to dephosphorylation of the enzymes present inside the cell. Now tell me one thing. You want to do glycolysis. Do you want upregulation of glycolysis or down regulation? You want to do more glycolysis in the liver. Do you want, beautiful answer, do you want more insulin to happen? Do you want more insulin to come? Why, sir? Can you understand now why I need insulin? Because what will insulin do, guys? A beautiful concept. This God is so beautiful. Once you bring the insulin on the surface of the cell, you will do dephosphorylation of the enzymes, guys. Because we know who is active in the dephosphorylated state. PFK1 is active. PFK2 is active in what? Dephosphorylated state. That means there should be another concept. Sir, we got your concept. If you want to convert this fructose 2,6-bisphosphate again to F6P, should you use a cleaving phosphatase enzyme? Should you use F26-bisphosphatase? I told you, if you want to make bisphosphate, you are using kinase. I have taught you already in the basic fundamentals of biochemistry. If you want to make bisphosphate, you are using the enzyme phosphofructokinase. But if you want to cleave this, what you will do, you will give the enzyme F26BPase, fructose 2,6-bisphosphatase. Fructose 2,6-bisphosphatase. If you want to produce fructose 2,6-bisphosphate, you have to activate the enzyme. You have to dephosphorylate the PFK2 enzyme. If you want to get back to destroy the F26BP, take the enzyme, remove the phosphate by the enzyme fructose 2,6-bisphosphatase. Fructose Bisphosphatase. Is it clear? Is it clear, friends? Guys, is it clear? Now listen to me. I came with insulin. Insulin came on the surface of the cell. It led to the dephosphorylation of the enzymes. And you know from the basic concepts that once I do dephosphorylation of phosphofructokinase 2, I will be making this enzyme active. Beautiful concept. That means if I do phosphorylation of this enzyme, I will be making this enzyme active. So PFK2 is active in the dephosphorylated state. Fructose 2,6 bisphosphonate is active in the phosphorylated state. True or false? I am talking about these two enzymes, guys. PFK2 is active in dephosphorylated state, and fructose 2,6 bisphosphatase is active in the phosphorylated state. That is, you have to add more phosphates, phosphate. Now what will happen? The dephosphorylation of PFK2. Now PFK2 is active, and that will start activating what? That will increase the production of F26 bisphosphate. This will in turn activate phosphofructokinase 1. Can we understand this? This will in turn activate the phosphofructokinase 1. Now you will be finally getting what? What? This F6P will be converted to fructose 1,6 bisphosphate. That's why what is the proof? If someone asks you a question, what is the proof that insulin is doing glycolysis? Give him this example. How you will give him this example? See how you will explain him. You will say, see my brother doctor, see my colleague, see my dear. Once insulin comes on the surface cell receptor, it will lead to dephosphorylation of the enzymes, because we know from the concept that phosphofructokinase 2 is active in the dephosphorylated form, while as fructose 2,6 bisphosphatase is active in the phosphorylated form. What will finally happen is you are giving insulin, dephosphorylation of enzyme is happening, PFK2 is activated. Once you have PFK2, more PFK2, let me show you the diagram, whatever I am speaking should not get lost. Once I have activation of PFK2 will lead to more production of F26BP, and we know fructose 2,6-BP is the positive allosteric inducer of phosphofructokinase 1. This phosphofructokinase 1 will convert more amount of fructose 6-phosphate to fructose 1,6 bisphosphate. Finally, you will be starting the glycolysis leading to the pyruvate. This is the reason insulin is increasing the glycolysis, guys. Is there any doubt in this? Is there any doubt in this enzyme? Can we help you? Can we help you something else here? Can we help you with glucagon? Can we help you with glucagon, guys? Guys, can we help you with glucagon? Guys, what's up? Guys are lost. I hope you understood insulin. Can I tell you the what was that? Colarly, isn't it? Colarly, or what was that? No, opposite. Glucagon. Now what will glucagon do? Now tell me one thing, guys. Do you want, is glucagon active in phosphorylated state or dephosphorylated state, guys? Glucagon, what does glucagon do? Phosphorylated state. Beautiful answer. Now see what will happen. Will the glucagon cause phosphorylation of the cellular enzymes? Will it cause phosphorylation of the cellular enzymes, guys? Will it cause phosphorylation of the enzymes? Who is active in the phosphorylated state inside the cell? Who is active in the phosphorylated state inside the cell from these two enzymes, pyruvate PFK2 or fructose 2,6 bisphosphatase? Who is active in the phosphorylated state? If glucagon is doing the phosphorylation, it is the fructose 2,6 bisphosphatase. So phosphatase, what will be having? So that means I will be having less amount of fructose 2,6. That means if I am phosphorylating fructose 2,6 bisphosphatase, that means I will be destroying more amount of fructose 2,6 bisphosphate. That means I will be decreasing the level of fructose 2,6 bisphosphate in the cytosol. That means I will be decreasing the positive inducer. That means I will be decreasing the inducer of PFK-1. Therefore, it means the levels of F26BP will start going on decreasing and decreasing, reducing and reducing, and whatever level is left, whatever has been produced, that is also being consumed. What will happen? You will have down regulation of phosphofructokinase 1. That means your phosphofructokinase activity will get decreased, guys. Is someone having any doubt with this? Glucagon phosphorylation. Who is active in the phosphorylation? Fructose 2,6 bisphosphatase. Who is active in the dephosphorylated state? Phosphofructokinase 2, active in the dephosphorylated state. Once you are giving glucagon, phosphorylating enzyme. Who is active in the phosphorylated enzyme? Fructose 2,6 bisphosphatase. What will you getting more? You will be destroying more amount of fructose 2,6 bisphosphate. Oh sir, fructose 2,6 bisphosphate getting destroyed. Oh, it was a regulator, positive regulator for PFK-1. But I am having less amount now. No regulator, no positive regulator. What will happen? Less amount of glycolysis. And that too, this guy ATase, phosphatase is killing more amounts of it's consuming fructose 2,6 bisphosphate at a high level. Finally will end in a decreased fructose 2,6 bisphosphate level in the cytosol will lead to decrease in the glycolysis. Glycolysis will stop. By the way, there is an interesting thing about this enzyme. You should respect this enzyme. You should respect this enzyme. What is it? It is a bifunctional enzyme, guys. Bifunctional enzymes. Bifunctional enzyme means if you have read about the basics of cell biology, I hope. If this is a peptide chain, what happens is I have two enzymes on the same peptide. Enzyme number 1, enzyme number 2. Enzyme number 1, enzyme number 2. PFK2, F26BPase. These are on the same peptide, single chain of polypeptide. But enzymes are at different places. So it is showing me a dual regulation. This is the beautiful enzyme on the same peptide, which is showing me the dual enzyme. That is, it is biphasic regulation. It is showing me biphasic regulation, guys. It is showing me biphasic regulation. So the state of affairs is that, the thing is that it will depend which activity you want. What a beautiful enzyme. It will depend what type of activity you want. Okay. So if it will depend what is activating them. Is it insulin or glucagon? What a beautiful enzyme. If you have insulin, he will say, okay, go for more amount of PFK2. He will say, go more amount of PFK2, dephosphorylated state. If you have having glucagon, he will say, express more amount of fructose 2,6 bisphosphatase. Express more amount of fructose 2,6 bisphosphatase. Fructose 2,6 bisphosphatase. That means if I am producing a positive action on PFK2, I should be inhibiting this action. That's why they made it a phosphorylation and a dephosphorylation. You should respect your phosphates. This is because how your life is sustained on this earth. If you had no phosphorylation, dephosphorylation, you would not be able to do glycolysis. Your body would not know. The body is in fasting state. You have less amount. You are hypoglycemic. You are having less blood glucose level. What will happen? They will start finding the alternatives. Glucagon will start increasing. Glucagon will start, they will start breaking your all storage forms of the glucose. And it's only because of this phosphatase enzyme, guys. Only because of its phosphatase enzyme. I hope you have enjoyed this lecture. This was all about the regulation. I thought, you get confused while you are preparing for a license exam or something, that, first year people also, that how insulin and glucagon are working. It's a beautiful concept that you can give the proof that insulin is a positive regulator of glycolysis. And I hope you have understood this topic very well. You should subscribe to this channel for more special videos coming on this channel. And stay safe in Corona. See you all tomorrow. Bye-bye.